opportunity to present our work. And today I will tell you a story about a population genomic analysis on Aedes aegypti that was conducted in my lab by Alejandro Nabor-Lozada Chavez and was recently published if you're interested. So the focus is Aedes aegypti. Aedes aegypti is the primary vector of arboviruses in subtropical and tropical regions of the world. And how the cycle works, it depends on the female's requirement for blood from a vertebrate host. So the females feed on a host that is infected, they pick up the virus and then they transmit the virus to a new host during a subsequent blood meal. Arboviruses are non-retroviral RNA viruses. They can have a single strand, double strand, single strand plus or minus genome, but they are RNA viruses. And because they are non-retroviral, they do not encode for reverse transcriptase and integrase, and they are supposed to spend their life cycle within the cytoplasm of the host cell. Unexpectedly, when we look at the genome of both Aedes aegypti and Aedes albopictus, which is another vector and is prominent in temperate regions of the world, we found that in their genomes there are hundreds of sequences of viral origin. And these viruses are non-retroviral RNA viruses. Here you see a screenshot, and each of these dots is a viral sequence, which we call them as NREVs, which stands for non-retroviral endogenous viral elements. And these dots are color-coded based on the viral families to which they have more similarities to, and you can see Flaviviridae and Rhabdoviridae, which are the families which contain most of the arboviruses. And these sequences are distributed on the x-axis based on the length. So you immediately understand that these sequences are short. So it's not the whole genome that gets integrated, are fragments of these viruses that gets integrated. And we look at where do these sequences land, and they don't land casually across the genome, but they are enriched in pRNA clusters where they are flanked by transposable elements. pRNA clusters are specific regions within the genome. They were discovered in 2007 in Drosophila melanogaster as regions which are composed of fragments of transposable elements which produce small RNAs called pRNAs, which act during germline formation to shut down transposable elements based on sequence complementarity. So this observation, the fact that these viral integrations are enriched in pRNA cluster, brought us to ask whether these pRNAs are also produced by viral integration. And we observed that both in Aedes aegypti on the bottom and on top, and Aedes albopictus on the bottom, when we map small RNAs to viral integration, the primary class of small RNAs that map to this viral integration are pRNAs. So this discovery were really the basis for the idea that we proposed to the human science frontier in association with Ronald van Rey from the Netherlands and Jamie Susaneto from Brazil, and we hypothesized that this viral integration constitute a memory of previous viral infection, and they can act to limit cognate viral infection. And this is a kind of groundbreaking because mosquitoes are invertebrates, so they are supposed to have only native immunity, not adaptive immunity, and this would be a mechanism of adaptive immunity. So a consequence of this hypothesis is that if this were to be true, then we would expect that if we take mosquitoes in the wild and we look at their genome, we would expect that the pattern of this viral integration changes, and it should correlate with viral exposure. So to answer this question, we actually collected 685 mosquito genomes across the world. Here I color-coded the states in which we got the mosquitoes for which we derived the genome, and we were particularly careful in choosing places in which arboviruses are endemic, so specifically Mexico, Brazil, and Thailand. And we checked in their genomes what is the pattern of viral integration. And with respect to the viral integrations that we had annotated in the reference genome. And here is a PCA analysis that shows how our samples, our populations separate with respect to the pattern of viral integration. And differently than what we expect, we saw that these mosquitoes are not, these populations are not separated depending on the one where there is higher arbovirus circulating with respect to not, but they separate based on their geography. So we see a group of out of Africa and group of African mosquitoes. And so this is not conforming to our hypothesis, but it is still relevant with respect to the biology of Aedes aegypti. Why? Aedes aegypti is a species which is native of Africa, and it is historically documented that Aedes aegypti left Africa specifically in a moment in history when the Atlantic slave trade started in the 1500s. And Aedes aegypti moved to the American continent, then to the Mediterranean region, then to Asia, and then to Oceania. So this is historically documented. Not only that, mosquitoes that are in Africa and out of Africa are morphologically different. In Africa, in Africa we have mosquitoes which are darker in abdomen, and they're called Aedes aegypti formosus or AAF. And in out of Africa we have Aedes aegypti, which are lighter in color. But more important than their morphology is their behavior. These mosquitoes behave differently. Aedes aegypti formosus are generalist, meaning that females feed indistinguishably on humans and animals, and they are sylvatic, so the females tend to deposit their eggs in sylvatic environments. On the contrary, Aedes aegypti have been adapted to human environments, so they prefer to feed on humans, and they prefer to lay eggs in artificial containers. And because of this behavior, this process of self-attraction to humans is called self-domestication. So it's a domestication process which is not human-mediated, but it's a natural self-selective process of association with human dwellings. And this is extremely significant because of these behaviors, Aedes aegypti have a higher vectorial capacity than Aedes aegypti formosus, so they are more epidemiologically relevant. So because of this reason, and because we saw this separation in the pattern of viral integration, we said, okay, we are in a good setting. Let's use this data to look at SNP actually and see if we can use these sources to actually understand whether we can see a signature of this self-domestication, of this behavioral differentiation between Aedes aegypti formosus and Aedes aegypti. And indeed, we annotated 1.5 million non-redundant biallelic SNP in our samples. We verified that our samples separate between Africa and out of Africa, with the exception of these mosquitoes here. And these mosquitoes are specific. These mosquitoes are mosquitoes which were collected in Rabai, which is a location in Kenya. And in this location, Aedes aegypti was identified. So how can we explain the presence of Aedes aegypti in Rabai? We did a phylogeny tree considering all our mosquitoes, and you see that Rabai mosquito land here. So they land within Aedes aegypti, and they land next to mosquitoes from Jeddah in Saudi Arabia. So the hypothesis is that Rabai mosquito represent a back to Africa event of Aedes aegypti that went back to Africa. But all of our samples, all of the other samples are actually all African or out of Africa. So we are really in the position to use this data to ask the question, what is the signature that differentiates the genome of Aedes aegypti formosus and Aedes aegypti? And to do that, we used three different approaches. We look at signature of strong selective sweep using rates. We look at outlier SNPs that would correlate with Aedes aegypti and Aedes aegypti formosus with a higher probability with respect to drift alone using PC-Adapt. And then we look at the direction of selection statistics through the McDonald and Craigman test. And in the Venn diagram, you see the number of genes that were identified, that had outliers that were identified by the three methods, and what we call as AAA, molecular signature genes, so signature of adaptation in Aedes aegypti, are genes which had signature of adaptation by at least two of these methods. And we identified 186 of these genes. And what is the functions of these genes? These genes are associated mainly to chemosensory, metabolic, neuronal function. And among the chemosensory gene, there are some important culprits like GR1, OR8, and ER8A. And why they are important? Because these genes have already been associated to mediate perception of CO2, amine, and lactic acids, which are stimuli that drive the preference of Aedes aegypti towards humans. But what these results tell you is something a little bit broader than the context of mosquitoes, because I told you that what we observe in Aedes aegypti is a phenomenon of self-domestication. So it's a natural process of attraction and adaptation to humans. When we think of domestication, we don't think of self-selective domestication. What we think most is the other process, the human-driven process of control over the reproduction and the survival of animals. And this has been happening since the Neolithic time with animals such as pigs or rabbits, but also insects like the Bombyx mori, which produce silk. And when people look at signature of human-driven domestication in these animals, so across different phylogenetic scale, vertebrates and invertebrates, they saw that the genes that are associated with domestication are mostly associated with the fine-tuning of neuronal and metabolic function, like we observe in mosquitoes. So our results suggest that the signature of domestication, where they be human-driven or self-selective, is similar and depends on the fine-tuning of metabolic and neuronal network. Throughout my talk, I talk about domestication. I didn't tell you very much about viral integration. Through this approach, we didn't really prove if a viral integration have any antiviral activity or not. So we can hypothesize that viral integration can still have antiviral activity, even if we don't see a correlation in the pattern of viral integration across the populations. And we have to think a little bit more about the viral integration. And I told you that these viral integration are enriched in pRNA cluster, but we saw also some which are not in pRNA cluster. And the ones that are not in pRNA cluster, we observed that they are fragmented sequences, but they span a complete viral open reading frame, a complete viral gene. And so the hypothesis is that they can have been co-opted by the host for immunity functions, like it has happened for integrations from other viruses, and this is something that has been already observed. The other possibility is that when we look at the pattern of viral integration, we check at the viral integrations that we had annotated in our genome. But what happens in the field is that mosquitoes can pick up novel viral integration that are not present in the reference genome, but they're present in the wild. Okay, so we have to distinguish these new potential viral integration and novel one. So regarding the first question, we are using a cell-based approach to try to answer this question. We identify some viral integration that are outside pRNA cluster that are transcribed through by transcriptomic analysis. We observe that, and we're using a cell line approach. And just briefly what I'm showing is that when we tag one of these viral integration with GFP, we transfect a cell and then we infect them with a virus. We are using a self-using agent virus, which is a flavivirus, but it is an insect-specific virus, so it infects only insects. We see in presence of this viral integration, we see a decrease of self-using agent virus. For the second approach, for the second question, we had to develop bioinformatic tools to identify these novel viral integration, which you can think of them as large integrations. And by doing that, yes, we can find that field-collected mosquitoes have novel viral integration, and us and also a group in the Institute Pasteur demonstrated that the presence of this novel viral integration actually causes a reduction of cognate viral infection, but a reduction, not a clearance of viral infection, okay? And in a tissue-specific manner, not throughout the mosquito body, only in the ovaries. So as a summary, I showed you that some viral integration, not all. So the big question is why the mosquito have so many viral integration. And the Eve-mediated antiviral effect exists. It can exist in two flavors. But I also showed you that with this type of data, we can do even more than that. And we can ask a fundamental question on the evolution of the mosquito. And with that, I would like to thank, obviously, the Human Science Frontier for giving me the opportunity to explore this new fascinating research on viral integration and the additional fundings of my lab. Thank you very much. Thank you.